Hello viewers, welcome to this particular lecture on nitrogen fixation. In this particular lecture, I shall be discussing the different mechanism by which nitrogen fixation is brought about. Actually, I shall be discussing the different genes involved in nitrogen fixation, so we can very well call it molecular mechanism of nitrogen fixation. The major objectives of this particular lecture are to understand the mechanism of nitrogenase action, to know the different factors involved in nodule formation, we call it nod factors, and of course the nod genes responsible for nodule initiation. And towards the end, I shall be discussing the different Nif gene and their function and how they activate the nitrogenase enzyme. Now the complete equation of nitrogen fixation is like this. In presence of 16 ATP, nitrogen is converted to ammonia, and actually it also involves the reduction process. So there is equal involvement of 8 H plus ion and 8 electron. And this entire activity is actually brought about by a unique enzyme which is called nitrogenase. And nitrogenase, actually the molecular structure indicate it is formed of three different components, that is the Fe protein, and the Fe protein remain on both sides, whereas towards the center there is the MoFe or molybdenum iron protein. One of the Fe protein, which is produced by Nif H, has the bound nucleotide and it has 4 Fe and 4S center, while on the other side is also having 4 Fe and 4S, but it is produced by Nif H gene activity. But the most important component of nitrogenase is the central MoFe protein, which is produced by the Nif D and Nif K, and it is having 8 Fe and 7S, that is iron sulfur center. And definitely they are entirely involved in reduction of dinitrogen or nitrogen gas to ammonia. Now let us discuss how this particular nitrogen is reduced to ammonia. At the first place, the reduced ferredoxine is oxidized, that is, it will liberate out the electron which will be accepted by reduced or Fe protein, which will get reduced in presence of ATP, and that will be oxidized. And at the last step, this reduced MoFe protein is produced, which will be oxidized, but in the process nitrogen is converted to NH double bond NH, that is NH-NH molecule, and this molecule will follow two separate steps of reduction, and finally it will produce NH2NH2 and then finally NH3. So what we see is it is a stepwise reduction from nitrogen to ammonia, and the most important component is the M or the FeMO cofactor. This M will bind with nitrogen and will produce the diazenido compound, and this diazenido compound, that is Mn double bond NH, will produce two types of compound. On one side it is the nitrido or NM complex, and on the other hand it will produce the MNH-NH2 molecule. The nitrido compound will produce the imido, that is MNH compound, while the hydrazido compound will produce the MNH2NH2. At this point, they will liberate ammonia and combine to produce the amido group, which is MNH2. And finally, this MNH2 will produce the ammonia and M will be recycled. So what we see is a stepwise reduction, and in each step ammonia is produced. And more the ammonia is produced, naturally we say more nitrogen is fixed, because this ammonia will be utilized for the production of amino acids or protein. Now, there are different nitrogen fixing bacteria. Broadly, they are classified as aerobic, anaerobic, legumes, and of course mixed bacteria present in other host other than legume plant. The aerobic bacteria include Azotobacter, Bezerenkia, Klebsiella, of course not all of them, some of them are aerobic, and the cyanobacteria, this is also some of them are aerobic in nature. But the cyanobacteria, they do contain chlorophyll A, and so they are photosynthetic in nature. The anaerobic bacteria include Clostridium, D-sulfovibrio, purple sulfur bacteria, purple non-sulfur bacteria, both of them being photosynthetic, along with green sulfur bacteria, which is also photosynthetic in nature. We also have the leguminous bacteria. This is the rhizobium, and it is producing the nodule inside the root of legume plant. And we have the Frankia and Azospirillum, which is active in other plant like in cereals or in alder. Nitrogenase is the most important enzyme for nitrogen fixation, but the fact of the matter is that nitrogen activity or nitrogenase activity is only occurring in absence of oxygen. So different organisms adapt to different types of evading mechanism by which they evade high oxygen concentration. Now let us discuss one by one. In case of the free-living nitrogen fixing organism, which is the Azotobacter, the oxygen scavenging strategy include the production of extracellular polysaccharide or EPS. And this EPS, it is clearly shown that it increases in presence of oxygen. When the oxygen concentration is high as 20%, naturally the EPS thickness will be more. In case of rhizobium, there is a separate mechanism, and this is leghemoglobin. As the nitrogenase gets activated, the leghemoglobin will bind with oxygen and will take out oxygen from the nodule site, as a result of which the nitrogenase can be active without any oxygen's presence. In the third one, that is in filamentous cyanobacteria or blue-green algae, there is a specialized cell which is actually the heterocyst. And this heterocyst has some specific molecule in the wall, the HEP and the HGL or the honeycomb structure, which will prevent entry of oxygen. Not only that, cyanobacteria being a photosynthetic bacteria, they do not carry out the PS2, which is involved in oxygen liberation. So naturally only PS1 is involved in photosynthetic light phase, and so oxygen concentration remains minimum. So here we can see that this particular heterocyst, the oxygen is not allowed, but the nitrogen enters. The nitrogen is converted to ammonia in presence of nitrogenase on one side, and on the other side it is actually the production of glutamate, which is via Gogat reaction. This glutamate will produce the glutamine, and this glutamine will be operating in other cells producing protein. But most interestingly, sucrose will enter the heterocyst, and this will produce the glucose, and this glucose will be operating only via PS1 photosynthetic process, as a result of which this particular molecule will actually operating without oxygen concentration. Let us discuss the different aspects of nodule formation, which is important because it is the site of nitrogenase. Actually, the nodule formation is initiated by the nod factor. There are different nod factors which are produced by colonizing bacteria. Say for example, ideally the Sinorhizobium meliloti, which is actually responsible for nitrogen fixation in Lotus. The nod factor consists of 4 glucosamine unit of 16 carbon with 2 unsaturated bond, and there are actually an acetyl group at the non-reducing terminal sugar denoted by nod L, and a sulfate group which is at the reducing terminal sugar denoted by nod H and nod P and nod Q. So ideally it is a polysaccharide, and this nod factor is produced by different nod genes. They are called nod, NOL or NOE genes, and this expression of these genes will be there whenever there are a specific flavonoid produced by the plant. So the plant root produces flavonoid which is activating the nod factors, and as I told you, the nod factors are in common word lipocyto-oligosaccharides, and they have this typical oligomeric backbone of 4 oligosaccharide or 4 residue oligosaccharide, and the number may vary between 3 to 5, though most of them are 4. Now the NOT factor has to be received. Who is receiving that? It is received by the leguminous plant root, and it is received by the lectin. The lectin may be termed differently. It may be the PSL1 in pea or LE1 in soybean, and these NOT factors are recognized by Lys-M or Lys-M receptor and receptor kinase like NFR1 and NFR5 as denoted in Lotus japonicus. Sometimes in Medicago truncatula there are specific NOT factor binding protein which include the NFBS1 and NFBS2. Similarly, in case of Dolichos biflorus or glycine max, there is the typical lectin nucleotide phosphohydrolase or LNP responsible for NOT factor binding. And as I told you, NOT factor recognition and binding only occur in presence of the specific flavonoids produced by the plant root. So, if we discuss the different steps of nodule formation, it is like this. There will be interaction of flavonoid with the NOD factor protein, and there will be induction of nodulation gene. It is followed by the specific increase in NOT factor plethora, and then there will be initiation of nodule formation in the cortical region. So there is modification of the cell, and then there will be activation of the nitrogen fixing gene, and nitrogen fixation will be initiated. In case of Medicago truncatula and Sinorhizobium meliloti, there is a typical association. This can be explained by some specific gene expression, like step 1 is called nodule primordium formation and step 2 is root nodule differentiation. What happens is, it is clearly seen in this figure that the bacteria induce root hair curling and infection thread formation because of the expression of a particular gene called NOD40, and there will be some initiation of cell division marked by yellow cells, and the cell gradually will be losing their identity, and there will be formation of the primordium in inner cortex, and subsequently there will be two factors, that is ENO-D12 and ENO-D40. The outer cortical cells gradually will be modified to produce pre-infection thread. At this point, first stage is completed. In the second stage, the cells of the nodule primordium obtain their final identity. At the base of this primordium, there will be a central pink tissue differentiation, and this pink color is due to the formation of Leghemoglobin, which is equally important because the oxygen has to be taken away. And finally, there will be expression of the NOD12 and there will be maturation of the nodule, and the process gradually becomes mature. Now, there are different genes which are responsible for nitrogen fixation or induction of nitrogen fixation in different host plant. Some of these major genes include, in Lotus japonicus there is the NIN, which is actually inducing the curling of root hair. In Medicago sativa this is the MNN1008, which is actually helping in NOT factor initiation. In case of Medicago truncatula, the DMI1, 2 and 3 and HCl, different factors are there which are actually promoting the NOT factor activation. There is also the typical NSP, which is gradually helping in the differentiation of the inner layer, and the last one, that is the Pisum sativum, it has got the SIM set of genes, SIM2, SIM8, 9, 10, etc. This particular SIM genes actually control the infection thread formation, and they also help in calcium ion oscillation and definitely after the nodule formation is over, this activity is stopped. Now let us understand the molecular mechanism by which the nod factor is recognized. And of course here also I shall be considering the host plant as Medicago trunculata and the Sinorhizobium meliloti. So the Sinorhizobium meliloti is producing the specific nod factor, which is recognized by different receptors belonging to the Lis-M family, and that include the MT-NFP or MT-LYK3 receptor, and this particular reception is also mediated by the DMI2, which is actually a leucine rich repeat receptor kinase, and activation of this DMI2 will help in the curling of root hair. But in the downstream there is another one, which is DMI1 present on the surface of the nucleus, and this DMI1 is activated because of the nod factor reception, and it helps in calcium ion spike, and the spiking of this calcium ion will bind with calmodulin, and there will be activation of a specific calmodulin dependent protein kinase, and this will active two Grass family transcriptional regulator called LSP1 and NSP2, and that finally will bring about the nod factor mediated inner transcriptional change, which will set the stage for nitrogen fixation. It is not only the specific nod factor that is helping in the formation of nodule. Sometimes there may be mycorrhizal signal, like nod factor mediated by SIM10, or along with SIM10 if there is some mycorrhizal signal, it will activate DMI1 and DMI2 and SIM8 or SIM19 to produce the calcium spike, and this calcium spike may be inhibited if there is excess of ethylene. So ethylene concentration, if it increases, then nitrogen fixation will go down. So ethylene, being a gaseous plant hormone, is inhibitory to nitrogen fixation or nodule formation. But if ethylene concentration is under control, there will be the third DMI receptor active, which is the DMI3, and finally there will be ENO D12 activation, and there will be curling of root hair, and finally nodule formation. Ideally, this nodule formation is brought about by the expression of all these genes, that is the DMI1, 2 and 3. Of course, if there is nodule formation, the nodule formation is up to certain limit, because if this limit is not maintained, it will be a cancerous growth and that will block the plant process. So naturally, whenever the nodule formation is complete, automatically there will be a feedback mechanism by which the excess growth of the nodule will be arrested. So there are some pathways which are blocked after nod factor initiation, and that include the NSP or nodulation signaling pathway as denoted in Medicago truncatula, or the NIN or the nodule inception pathway as denoted in Lotus japonicus. So definitely we understand that plant, they have their own control mechanism by which excess growth of the nodule can be controlled. So now the house is set. It is set for nitrogen fixation, and when I say nitrogen fixation, there will be activation of the next set of gene, and that is the Nif gene. So what are Nif gene? The Nif genes are genes encoding enzymes involved in the fixation of nitrogen from the atmosphere, and of course the different Nif genes will be helpful in the production of different components of nitrogenase complex. The Nif genes were discovered in the 80s by two persons, that is the Gary Ruvkun and Sharon Long. They were responsible for characterization of different Nif genes and the activity in Nif operon. So as I told you, Nif gene regulation is a very important factor, because here also there will be initiation of Nif gene which will take place in low nitrogen concentration, and then when there is excess of nitrogen fixed in the form of ammonia, naturally their process has to end, switching off this Nif operon. So what we see is initially there will be NTRA activation, and this NTRA alpha subunit will produce the specific RNA polymerase that will operate the Nif operon, and nitrogen fixation will be initiated. And the nitrogen concentration will grow, and then what happens is there will be activation of some other genes, that is the NTRC, and this NTRC actually will block the process by Nif-L activation. So on one side it is Nif-A which is initiating nitrogen fixation, and on the other side it is the Nif-L which is inhibiting nitrogen fixation. So we have the fire and the water on each hand. On one side the fire, that is the Nif-A, will initiate nitrogen fixation, and after adequate nitrogen has been fixed, the water will stop the process in the process of Nif-L. But as I told you, Nif gene is not the question of Nif-A and Nif-L. There are different types of Nif genes which were discovered, and in Klebsiella pneumoniae only there are different types of Nif gene like the Nif Q and B, which is mainly responsible for the production of the phemocosynthesis. The Nif A and L, as already told, are the initial regulator of the nitrogen fixation process. The Nif F is responsible for the production of flavoprotein, and Nif MZW, VSU are responsible for producing different subunits of dinitrogenase reductase or the metallic center of the enzyme. Nif XN and E are responsible for pharmacobiosynthesis. Similarly, Nif YT, KDH are responsible for pharmacomaturation or dinitrogenase activity and also nitrogenase accessory protein production. And finally, it is the Nif J which is responsible for flavodoxin production. So what we see is the different types of Nif genes producing different components of nitrogenase, and that makes the nitrogen fixation process perfect. As because pharmaco is the most important center, we can clearly see multiple genes are responsible for the initiation, activation and functioning of the pharmaco center. Now there may be some other ways by which this Nif gene regulation can be brought about. As in case of Rhodospirillum rubrum, which is a free-living anaerobic photosynthetic bacteria, there is of course the normal Nif gene, but along with Nif gene, there is a unique feedback mechanism. And how this is brought about? It is brought about by ADP ribosylation of a specific arginine residue in the nitrogen complex. So when enough nitrogen has been fixed, the ribosylation will take place and it will actually stop the electron transfer, and thereby it will inactivate the nitrogenase activity. And the catalysis of this ribosylation is brought about by two genes, which are DRAG and DRAT. In case of another unique bacteria, which is Rhodobacter capsulatus, which is a free-living anaerobic phototrope, the Nif gene expression is following the same pattern as Nif-A like in Klebsiella, but here the NTRC which is activating Nif-A is different from that NTRC of Klebsiella. So we see it is the different form of NTRC which is bringing about different activity, but the activity broadly is activation of Nif-A or initiation of nitrogen fixation. So in this particular lecture, what I have discussed is, I have discussed the process of nitrogenase formation, I have discussed the structure of nitrogenase, I have discussed the different ways by which oxygen can be scavenged from the site of nitrogenase. Then I discussed about different knot factors, how these knot factors are recognized, how they become active and they produce a successful nodule. I have also discussed how this particular nodule is limited, that is the growth of this nodule is limited, And finally, I have discussed about the Nif gene or the nitrogen fixation gene, the different types of Nif gene, their function and how they bring about nitrogenase activity. And then towards the end, I have discussed the different types of Nif operating system operating in different bacteria. Thank you.